Welcome, curious minds, to another deep dive. Today, we're embarking on one of biology's most fundamental and, frankly, astonishing journeys.
We really are.
We're talking about the intricate process from conception right through pregnancy and into lactation. In this deep dive, it draws its core insights from a chapter summary of Boron and Boulpaep's Medical Physiology, which is, a truly foundational text.
It absolutely is. It's dense, no doubt, but incredibly rewarding. Our mission today is really to transform that rich material into clear, accessible understanding for you.
Okay.
We'll unpack the precise steps of fertilization, the remarkable transformations a body undergoes during pregnancy, and the fascinating physiology behind milk production.
And connect it all back.
Exactly. We'll connect these complex ideas to real-world clinical insights, making sure you walk away feeling, well, confident about this vital material.
Think of this as your shortcut, maybe, to understanding everything from how a single sperm finds an egg to the incredible hormonal symphony orchestrating 9 months of development and, beyond.
Let's dive in.
Here's where it gets really interesting. Let's start at the very, very beginning. How do the egg and sperm actually meet? It's far more orchestrated than you might imagine.
Yeah, it really is. So after ovulation, you have the fimbriae of the fallopian tube. They're these delicate sort of finger-like projections.
Yep.
They gently sweep over the ovary to collect the oocyte. And this oocyte, it isn't just a naked cell. It's protected by surrounding layers of specialized cells.
It's like a delicate package being carefully scooped up. Then what propels it on its journey gets it moving.
Exactly. Once it's inside the fallopian tube, tiny hair-like structures called cilia, along with rhythmic contractions of the smooth muscle in the tube wall,
Right.
they work together in concert to gently move this whole egg complex towards the uterus. It's a beautifully coordinated transport system.
Now, for the male contribution, a man can deposit, hundreds of millions of sperm?
That's right.
But how many actually make it to the specific region of the fallopian tube, the ampulla, where fertilization usually occurs?
It's astonishingly few. Only a tiny, tiny fraction we're talking about, maybe 50 to 100 out of hundreds of millions.
Wow.
And what's truly remarkable is how quickly they arrive, often within just 5 minutes or so.
5 minutes. Their own tail-powered swimming alone can't possibly account for that kind of speed?
No way. So
Yeah.
it's not just their sheer willpower or tail power at work.
Right.
Precisely. They get a major assist from the female reproductive tract itself. Forceful contractions of the uterus, cervix, and fallopian tubes. Often enhanced by factors like prostaglandins in the seminal fluid, they act like an internal current, propelling sperm rapidly into the upper reproductive tract.
Okay, so they get a push. But before a sperm can even penetrate the egg, it needs a sort of final awakening or maturation step, right, in the female reproductive tract.
Yes, that's crucial. This physiological change is called capacitation.
Capacitation.
It's where sperm gain the actual ability to fertilize an egg. Essentially, a protective protein coat on the sperm's surface is modified or removed.
Like getting ready for action.
Exactly, preparing it for the intense interaction with the egg. It's like shedding an unnecessary shield to become battle-ready.
Right.
And what's fascinating here is that this process isn't strictly confined to the body. It can also occur outside, like in lab settings for procedures, like in vitro fertilization, IVF.
Interesting. Okay, with capacitation complete, let's get into the main event. Fertilization itself. The egg is waiting. How did you put it?
In a sort of suspended animation, yeah. If it's not fertilized, it simply degenerates.
Right.
But when that crucial contact happens, it awakens.
So walk us through the precise sequence. It sounds like a dance.
It really is a complex but precise ballet of cells. So first, the sperm binds to the egg's outer jelly-like layer, the zona pellucida.
Okay.
It specifically recognizes a key protein, ZP3, on its surface. That kicks things off.
Then what?
Next, the sperm undergoes the acrosomal reaction. You can picture the acrosome as this specialized cap on the sperm's head. This cap bursts open, releasing powerful enzymes. These enzymes are absolutely crucial for dissolving a pathway through that zona pellucida.
Okay, so it enzymes its way through.
Partly. Once the path is sort of cleared, the sperm head rapidly oscillates, almost like a drilling, physically pushing its way through this outer layer.
Wow. Okay, it's through the zona. What now?
Then the membranes of the sperm and egg actually fuse. The sperm's genetic material enters the egg.
But the membrane stays behind.
Exactly. Its outer plasma membrane is left behind. Then, this penetration triggers a vital protective mechanism in the egg called the cortical reaction.
Cortical reaction. Sounds important.
It is. The egg releases enzymes that harden the zona pellucida, immediately preventing any other sperm from entering. This is how polyspermy fertilization by multiple sperm is avoided.
Ah, that makes sense.
It's a critical evolutionary safeguard because polyploid embryos, those with extra sets of chromosomes, almost never develop normally. And that same signal, the sperm's entry, also prompts the egg to complete its final stage of meiosis, its cell division, which it had paused mid-process.
So it finishes its development.
Right. And finally, the genetic material from both the sperm and the egg, the pronuclei, they come together, forming a brand new cell, the zygote.
The zygote.
This fusion, called syngamy, marks the true beginning of a new individual, and it's also when chromosomal sex, XX or XY, is determined.
What an incredible, precise series of events. It really highlights just how many things have to go right, perfectly timed, for conception to even occur.
Absolutely. So many potential failure points.
So we have a zygote. It's been formed, but its journey is far from over. What happens next as it seeks a home, a place to implant and grow?
Right, so the ovum is fertilized way up in the fallopian tube, usually the ampulla, but the newly formed conceptus, as we now call it,
Conceptus.
it stays there for about 72 hours, 3 days. During this time, it begins to divide rapidly, forming a solid ball of cells that looks like a tiny mulberry. That's called the morula.
Morula. And it's getting nutrients.
Yeah, it's nourished by secretions from the fallopian tube itself.
And meanwhile, the uterus is preparing for its arrival, right? Getting the gastroom ready.
Exactly. The uterus undergoes remarkable changes, preparing its inner lining, the endometrium, to receive and nurture this developing embryo.
So the morula travels down.
Yes. It then rapidly moves into the uterine cavity, and once there, it transforms into a blastocyst.
Blastocyst. What does that look like?
Picture a hollow, ball-like structure. It has an outer layer, the trophiectoderm, which will eventually form the placenta and other supporting structures.
Okay.
And inside, attached to one side, is the inner cell mass. That's the part that will actually develop into the embryo itself.
So it reaches the uterus as a blastocyst. Does it implant right away?
No, it actually floats freely in the uterus for a bit longer.
Really? How long?
For another 72 hours or so. So if you do the math, implantation normally occurs about 6 to 7 days after ovulation.
Wow, that long.
Yeah, and this precise timing, the specific window of opportunity, is absolutely crucial. Both the embryo and the endometrium have to be perfectly synchronized and prepared for that moment.
So what specific changes does the uterus undergo, this preparation process?
This transformation is called decidualization. It's driven mainly by progesterone. The endometrium gets thicker, much more vascularized, and its glands swell up with nourishing secretions.
Okay.
If conception doesn't happen, these changes just regress, and that leads to menstruation.
Right.
But in pregnancy, these changes are not only sustained, but dramatically enhanced. This specialized endometrium of pregnancy is called the decidua.
The decidua.
It's essential. It provides nutrients, anchors the embryo, and helps establish that vital connection.
And before it's fully implanted, while it's floating, how does this tiny blastocyst get nourishment?
From those very uterine secretions, progesterone is key here again. It stimulates the endometrial glands to produce this rich, nutrient-filled fluid, kind of like a nourishing soup.
A soup. What's in it?
Well, vital proteins, cholesterol, adhesion molecules, other essential factors, everything the blastocyst needs in those early days.
I've heard about these temporary, sort of finger-like projections on the uterine lining called pinopods. Are they important for implantation?
They are indeed. Pinopods are small, transient protrusions on the endometrial cells. They appear precisely around the time the embryo arrives, typically between day 19 and 21 of the cycle.
And what do they do?
They play a critical role. They seem to absorb uterine fluid, which actually helps bring the embryo and the uterine lining physically closer together. That proximity is vital for the initial attachment, and their formation is highly dependent on progesterone.
Okay, so the uterus is ready, the blastocyst is there. How does the physical implantation process happen? You said it wasn't just passive embedding.
No, it's quite an active process. First, the blastocyst has to hatch. It sheds that outer shell, the zona pellucida. Then it goes through 3 key stages.
3 stages.
First is apposition. That's just the initial loose contact between the blastocyst, specifically its outer layer, the trochectoderm, and the uterine lining, often in a little crypt or fold.
Just bumping up against it.
Pretty much. Then comes adhesion. This is where the blastocyst firmly attaches. It involves intricate molecular handshakes between the embryo and the uterine cells. Think specialized proteins called integrins that act like Velcro, helping the blastocyst bind strongly to the uterine lining matrix proteins like laminin and fibronectin.
Okay, so it sticks. Then the active part.
Then there's invasion. This is where it gets really dynamic. The blastocyst's outer cells, the trophoblast, they rapidly multiply and differentiate into two layers. An outer one, the syncytiotrophoblast, is key here.
Syncytiotrophoblast.
Yes, it sends out long, finger-like projections that actively penetrate and break through the uterine lining cells. They secrete enzymes like TNF-alpha and proteases that essentially degrade the surrounding tissue and basement membrane, allowing them to burrow deeper into the uterine wall, the stroma.
Wow, it actually digests its way in.
In a controlled way, yes. It's actively invading to establish a connection. And the uterine cells around it transform further into decidual cells, full of nutrients, which actually break down to feed the embryo initially. These projections reaching towards the mother's blood supply are the very beginning of the placenta's villi.
That's an incredible transformation, really, a dialogue between the embryo and the uterus.
It's a very active dialogue.
So the placenta, it's often called the baby's lifeline. How does this vital organ actually form and function after that initial invasion?
Right after implantation, these fluid-filled spaces, or lacuna, develop within that syncytiotrophoblast layer.
Okay.
These spaces quickly erode into the mother's blood vessels, first veins, then arteries, establishing direct communication. So maternal blood starts pooling in these spaces.
Like little lakes of blood.
Sort of, yeah. Then, finger-like projections from the inner cytotrophoblast layer grow into these spaces. These are the primary chorionic villi. They become more complex, getting a core of mesenchymal tissue, secondary villi, and finally, fetal blood vessels develop within them, tertiary villi.
And that's the mature structure.
Essentially, yes. In the mature placenta, you have this vast, interconnected network of spaces filled with constantly flowing maternal blood, the intervillous space. Into this space, the mature chorionic villi protrude like a dense forest of trees branching out.
Okay.
Inside these villi, fetal blood circulates in capillaries, separated from the mother's blood by only a very thin barrier, just the fetal capillary wall, a bit of connective tissue, and the trophoblast layers. This design maximizes the surface area for exchange. It's incredibly efficient.
So how does the blood actually circulate? How does exchange happen?
Maternal arterial blood spurts into the intervillous space from spiral arteries in the uterine wall. It flows relatively slowly, cascading over the surfaces of all those villi.
Allowing time for exchange?
Exactly, allowing ample time for nutrients and oxygen to pass to the fetal blood, and waste products like CO2 to pass back to the mother before the maternal blood drains out through veins.
And the fetal side? How does its blood flow?
Fetal blood, which is deoxygenated and carrying waste, arrives at the placenta via two umbilical arteries. Remember, arteries carry blood away from the fetal heart here.
Right, opposite of postnatal circulation.
Correct. These arteries branch extensively within the chorionic villi, forming that vast capillary network for exchange. Then, the newly oxygenated, nutrient-rich blood collects and returns to the fetus through a single umbilical vein.
Fascinating.
Yeah.
What about the amniotic fluid the baby is floating in? What's its role?
The amniotic fluid serves two crucial purposes. First, it's a mechanical buffer. It cushions the fetus, protecting it from bumps and jolts, and gives it space to move and grow properly.
Okay.
Second, it's involved in waste handling. Initially, it's mainly derived from maternal fluid, but later in pregnancy, the fetal kidneys become the major source. Fetal urine basically makes up most of the fluid.
Fetal urine? Really?
Yep. And the fetus also swallows the fluid, so there's a constant turnover, processed via the fetal gastrointestinal tract, lungs, and the amniotic membrane itself. It's a dynamic environment.
Okay, back to the placenta. How do all the essential substances actually get across that barrier between mother and fetus? It's not just simple diffusion for everything, is it?
No, definitely not. The placenta is smart. It uses multiple transport mechanisms depending on the substance.
Like what?
Well, gases like oxygen and carbon dioxide, they move by simple diffusion down their concentration gradients. Same for lipid-soluble things like steroid hormones and waste products like urea.
Okay.
But glucose, which is vital fuel, needs help. It uses facilitated diffusion. Specific transporters carry it across.
Faster than simple diffusion.
Yes. And then for things the fetus really needs to concentrate, like amino acids for building proteins, vitamins, minerals like calcium and iron, those often move by secondary active transport. The placenta actively pumps them across, sometimes even against a concentration gradient.
Wow. What about bigger things like antibodies?
Great question. Large molecules like LDL cholesterol particles, iron bound to transfer in certain hormones, and crucially, maternal antibodies, specifically IgG, cross via receptor-mediated endocytosis. The placenta literally grabs them, engulfs them, and transports them across to the fetus.
And that maternal IgG is how the baby gets passive immunity, right?
Exactly. It provides vital protection in the first few months after birth before the baby's own immune system is fully up and running.
Let's talk about oxygen specifically. You mentioned the barrier is thin, but how does the fetus get enough oxygen when the maternal blood PO2 in that intervillous space isn't actually super high, maybe 30, 35 millimeter Hg?
That's a brilliant adaptation. The key is fetal hemoglobin.
Ah, HbF.
Right, fetal hemoglobin has a much higher affinity, a stronger attraction for oxygen than adult hemoglobin, HbA. So even at that lower maternal PO2, fetal hemoglobin can effectively pull oxygen away from the maternal hemoglobin and become highly saturated, maybe around 85% saturated in the umbilical vein blood.
So it loads oxygen more easily.
Precisely. Plus, the fetus has a higher cardiac output relative to its body size, and its hemoglobin concentration is higher later in pregnancy. It's a multi-pronged strategy to ensure adequate oxygen delivery.
And CO2 just goes the other way, down its gradient.
Pretty much, yes. There's a small gradient favoring CO2 movement from fetus to mother, and fetal blood has a slightly lower affinity for CO2 as well.
Okay, beyond transport, does the placenta make its own hormones? You mentioned HCG.
Oh yes, it's a major endocrine organ. As we said, human chorionic gonadotropin, HCG, is vital early on. It rescues the corpus luteum in the ovary, keeping progesterone levels high until the placenta can take over steroid production itself.
And is what pregnancy tests detect.
Exactly. HCG also seems to promote the growth of the trophoblast itself. Then there's another really important group, human placental lactogen, or HPL, also called human chorionic somatomammatropin, HCS.
HPL, what does that do?
HPL is structurally similar to growth hormone and prolactin. Its main job is to help coordinate the mother's metabolism to ensure a continuous supply of fuel, especially glucose, to the fetus. It makes the mother a bit insulin resistant, shifting her fuel use towards fats, which spares glucose for the baby.
So it manages the fuel economy.
Perfectly put. It also helps prepare the maternal mammary glands for lactation later on.
So the placenta isn't just a passive filter. It's actively managing the whole pregnancy environment. Transport, metabolism, hormones, an incredible organ.
Truly remarkable.
Okay, speaking of hormones, maternal levels of progesterone and estrogens, especially one called estriol, skyrocketed during pregnancy, right? Way higher than normal cycle.
Dramatically higher. And these elevated levels are absolutely necessary to maintain the pregnancy. Progesterone, for example, is crucial for keeping the uterus relaxed and preventing premature contractions.
So how does the body sustain these super high levels? You mentioned the placenta takes over from the corpus luteum.
Right. Early in the first trimester, it's the corpus luteum, stimulated by that placental HCG. But by about 8 weeks, the placenta becomes the main steroid factory.
Okay.
However, and here's where it gets really fascinating and integrated, the placenta can't actually produce all the necessary steroids entirely on its own. It needs help.
Help from whom?
From both the mother and the fetus. This leads to the concept of the maternal placental fetal unit. It's a true team effort.
A three-way partnership. So what are the placenta's shortcomings in making these steroids, and how do the mother and fetus compensate?
Okay, so the placenta is an imperfect endocrine organ in a couple of ways. First, it can't make enough cholesterol from scratch, which is the basic building block for all steroid hormones.
So where does it get cholesterol?
The mother supplies most of the cholesterol, primarily delivered as LDL particles in her blood.
Okay, that's the mother's contribution. What about the fetus?
The placenta lacks certain key enzymes. Specifically, it's missing 17-hydroxylase and 17,020-desmolase, which are needed to convert progesterone precursors into androgens, the intermediates for making estrogens like estrone and estradiol. It also lacks 16-hydroxylase, which is needed specifically for making estriol the main estrogen of pregnancy.
So 3 missing enzymes. Where do they come from?
The fetal adrenal gland and the fetal liver supply these missing enzymes. The fetal adrenal glands are surprisingly large at term, sometimes as big as an adult's kidney, partly because they're so busy making these steroid precursors.
Wow. But why this complicated setup? Why doesn't the fetus just make its own estrogens directly? Wouldn't that be simpler?
That's a brilliant question, and it highlights the elegance of the system. The fetus shouldn't be exposed to the very high levels of active estrogens that the mother needs to maintain the pregnancy. That could be harmful, especially for a male fetus.
Ah, okay. So how does the unit protect the fetus?
It uses several clever strategies. First, as we said, the fetus lacks the final enzymes, so it can't make the potent estrogens itself. Second, the placenta acts like a massive sink, rapidly converting any weak androgens the fetus does make into estrogens, preventing those androgens from building up and potentially masculinizing a female fetus.
Okay.
And third, the fetus takes many of the steroid intermediates it produces, like DHEA, and attaches a sulfate group to them. This sulfation makes them biologically inactive.
Like putting a safety cap on them.
Exactly. These inactive sulfated precursors then travel to the placenta. The placenta has an enzyme, sulfatase, that clips off the sulfate group, reactivating the precursor. Then the placenta uses its own enzymes to complete the conversion to the final active estrogens, which are then released primarily into the mother circulation where they're needed.
That is incredibly intricate. A beautiful division of labor to protect the fetus while supporting the mother.
Precisely. The maternal placental fetal unit is a masterpiece of physiological coordination.
So pregnancy isn't just about fetal development. The mother's body undergoes these profound whole-body transformations. What are some of the most significant adaptations we see?
The maternal body truly adapts in remarkable ways, largely driven by that hormonal surge and, of course, the physical presence of the growing uterus.
Let's start with the cardiovascular system. Big changes there.
Huge changes. Maternal blood volume increases significantly by as much as 45% in a single pregnancy, maybe even more with multiples.
45%. Why so much?
Well, plasma volume goes up more than red blood cell volume, leading to a slight dilutional anemia, which is normal. This expansion is crucial for meeting the increased metabolic demands of the uterus and placenta, providing adequate flow, protecting against potential problems with venous return when lying down, and importantly, safeguarding against blood loss during delivery.
What drives that volume increase?
It's largely hormonal. Estrogen and progesterone cause vasodilation, a widening of blood vessels, which decreases peripheral vascular resistance. This signals the kidneys to retain more salt in water, partly via the renin-angiotensin-aldosterone system. Aldosterone levels go way up.
Okay. And the heart itself?
Cardiac output, the amount of blood pumped by the heart each minute, also increases substantially. It goes up by 35–40% relatively early in the first trimester, peaking maybe 45% higher than pre-pregnancy levels.
How does it manage that?
Mostly through an increase in both stroke volume, how much blood is pumped per beat, and heart rate. And this increased output is directed strategically. Blood flow increases significantly to the kidneys, the skin, the breasts, and especially the uterus, which goes from receiving about 1% of cardiac output to maybe 15% or more near term.
Wow. With all that extra volume and cardiac output, what happens to blood pressure? Does it go up?
Interestingly, no, not usually. Despite the large increase in volume, arterial blood pressure typically decreases slightly during mid-pregnancy. Because of that significant fall in peripheral vascular resistance caused by the vasodilating effects of hormones like progesterone and prostaglandins, the system opens up, so pressure drops a bit. It usually rises back towards pre-pregnancy levels during the third trimester, but generally stays at or below normal.
And you mentioned posture matters.
Yes, especially in late pregnancy. When a pregnant woman lies flat on her back, supine, the heavy uterus can compress the inferior vena cava, the large vein returning blood from the lower body to the heart.
Oh, right.
This can significantly reduce venous return and, consequently, cardiac output. Lying on her side relieves that pressure, so cardiac output is often higher in the lateral position.
Good clinical tip. Okay, what about respiratory changes? Does breathing change?
Yes, significantly. Progesterone is a key player here, too. It acts as a respiratory stimulant.
How so?
It increases sensitivity to CO2 in the brain's respiratory centers, leading to increased alveolar ventilation. Basically, you breathe more deeply and slightly faster. The diaphragm also gets pushed up by the growing uterus, maybe by about 4 centimeters.
That sounds like it would make breathing harder.
You'd think so, but the ribcage actually widens to compensate, so total lung capacity doesn't change much. And airway resistance actually decreases, making airflow easier.
So overall, the mother is breathing more efficiently.
Yes. Tidal volume, the amount of air inhaled with each normal breath, increases markedly by about 40%. This significantly boosts alveolar ventilation starting quite early in pregnancy, maybe around 6 weeks.
And what's the effect on blood gases?
This increased ventilation causes maternal arterial PCO2, the level of carbon dioxide in the blood, to fall typically from around 40 millimeter Hg down to about 32 millimeter Hg.
So she's blowing off more CO2.
Exactly. This creates a mild respiratory alkalosis, a slightly higher blood pH. The kidneys compensate for this by excreting a bit more bicarbonate, bringing the pH back towards normal. This lower maternal PCO2 also helps create a gradient to pull CO2 away from the fetus across the placenta.
Clever. Okay, and of course the nutritional demands must change dramatically too.
Absolutely. Pregnancy significantly increases the need for calories, protein, vitamins, and minerals. For instance, an additional 30 grams of protein are needed daily, roughly, for building fetal and maternal tissues.
What about specific micronutrients?
Iron is a big one. This is really key clinically. Pregnancy requires a net gain of about 800 milligrams of iron overall. Most of this goes to expanding the mother's red blood cell mass to carry more oxygen, but a good chunk goes to the placenta and the fetus itself.
Can diet usually cover that?
Very rarely. Few women have adequate iron stores to meet this demand, and typical diets usually don't provide enough. So iron supplementation, about 60 milligrams per day of elemental iron, is almost always recommended during pregnancy.
Good to know. What else?
Folic acid is another critical one. Maternal folate requirements increase dramatically, needed for DNA synthesis and cell division, especially red blood cell production. Deficiency can cause maternal anemia, but more critically, it's strongly linked to neural tube defects in the developing fetus, like spina bifida.
Which are preventable.
Largely, yes. Folic acid supplementation, typically 400 to 800 micrograms per day, ideally starting even before conception, is crucial for prevention. It's a major public health recommendation.
Okay. All these changes, extra blood, uterine growth, fetus, placenta, must lead to significant weight gain. Where does all that weight actually go?
Right. For a woman starting with a normal BMI, a total gain of about 11.5 to 16 kilograms, that's roughly 25 to 35 pounds, is generally recommended.
And how much of that is the baby?
Surprisingly, less than you might think. The fetus itself accounts for only about 3.3 kilograms, or maybe 7.5 pounds, on average at term.
So where's the rest?
The rest is distributed among the placenta, about 0.6 kilograms, the amniotic fluid, 0.8 kilograms, uterine growth, 1 kilogram, increased maternal blood volume, 1.2 kilograms, breast tissue development, 0.4 kilograms, and necessary accumulation of maternal body fat and interstitial fluid, around 4 kilograms or more.
So it's all functional weight gain.
Exactly. And achieving adequate weight gain within the recommended range is strongly correlated with a favorable pregnancy outcome, healthier baby, fewer complications.
Okay, so after 9 months of all this incredible development and adaptation, how does the body know it's time for labor, for parturition to actually begin?
That's the million-dollar question, and we don't know all the triggers definitively. For most of pregnancy, the uterus remains largely quiescent, relaxed, likely kept that way by high levels of progesterone and perhaps hormones like relaxin.
Okay.
Then, in the last month or so, you often get these weak, irregular contractions, sometimes called Braxton-Hicks contractions, or like practice.
Right.
Labor officially begins when regular, rhythmic, and forceful contractions develop that lead to progressive cervical thinning that's effacement and dilation.
What flips the switch?
It seems to be a complex interplay of factors: endocrine signals, paracrine signals, local chemical messengers, and mechanical factors, like the stretching of the uterus and cervix by the growing fetus. And once labor is initiated, it's powerfully sustained and amplified by positive feedback loops.
What are some of the signals involved? Is it a drop in progesterone like in some animals?
In some species, like rabbits, yes, a clear drop in progesterone triggers labor. In humans, it's not quite so clear-cut. Progesterone levels don't plummet right before labor starts. However, progesterone is certainly crucial for maintaining pregnancy, and drugs that block progesterone can induce labor, so it plays a role in keeping things quiet.
What about signals from the fetus itself? Does the baby signal it's ready?
There's strong evidence for that. The fetal hypothalamic pituitary adrenal HPA axis seems to be involved. For instance, in rare human pregnancies where the fetus has anencephaly, a severe brain malformation affecting the HPA axis, gestation is often significantly prolonged.
Suggesting the fetus normally contributes a go signal.
Exactly. Cortisol from the fetal adrenal gland might be part of that signal. However, the most widely accepted key players in initiating and driving labor contractions are prostaglandins.
Prostaglandins again. They seem to pop up everywhere.
They do. They're synthesized right there in the uterus, the placenta, and the fetal membranes, amnion and chorion. They act locally on uterine smooth muscle cells, strongly stimulating contractions. And interestingly, oxytocin can actually boost prostaglandin synthesis in the decidual cells.
So what are their main effects during labor? What makes them so important?
They have three major effects, really. First, they strongly stimulate uterine smooth muscle contraction.
Okay.
Second, they potentiate contractions induced by oxytocin. They do this partly by promoting the formation of gap junctions between uterine smooth muscle cells.
Gap junctions, like in the heart.
Exactly the same principle. These gap junctions allow electrical signals and ions to pass directly from cell to cell, enabling the uterine muscle cells to contract in a coordinated, synchronous way. Much more effective contraction.
Makes sense. And the third effect?
Third, and crucially for starting labor, they cause softening, dilation, and thinning effacement of the cervix. The cervix needs to ripen and open up, and prostaglandins are key drivers of this process, which kind of resembles an inflammatory reaction locally.
And because of these effects?
Because of these effects, prostaglandins like PGE2 or PGF2 or their analogs are widely used clinically to ripen the cervix and induce labor when needed.
Okay, so prostaglandins are major initiators. What about oxytocin? The love hormone is also famous for its role in labor.
Absolutely. Oxytocin is a peptide hormone made in the hypothalamus and released from the posterior pituitary gland into the bloodstream.
And it acts on the uterus.
Yes, it binds to specific receptors on uterine smooth muscle cells. This triggers a cascade involving calcium release inside the cells, which ultimately leads to muscle contraction. Oxytocin also stimulates those decidual cells to produce more prostaglandins, amplifying the effect.
So does oxytocin actually initiate labor in humans?
That's debated. While giving oxytocin can certainly induce or augment labor contractions, the prevailing view is that maternal oxytocin release isn't the primary trigger that starts spontaneous labor in humans. Fetal oxytocin might play a role, as its levels seem to rise during the first stage.
But it becomes important later.
Critically important. As pregnancy progresses, under the influence of rising estrogen levels, the number of oxytocin receptors in the uterus increases dramatically, maybe up to 200-fold by the time early labor starts.
Wow, so the uterus becomes incredibly sensitive to it.
Exquisitely sensitive. So even if basal oxytocin levels don't rise dramatically before labor, the uterus is primed to respond powerfully when oxytocin release does increase during labor.
So it's more about sustaining and strengthening labor once it's underway, rather than kicking it off.
Exactly. Once labor is initiated, maternal oxytocin is released in pulses, and the frequency and amplitude of these pulses increase as labor progresses, especially during the second stage, pushing.
What triggers that release during labor?
The primary stimulus for maternal oxytocin release is distension or stretching of the cervix as the baby descends. This is known as the Ferguson reflex. Stretching sends nerve signals up the spinal cord to the hypothalamus, triggering oxytocin release from the posterior pituitary.
A positive feedback loop.
A classic positive feedback loop. Stretch oxytocin, stronger contractions, more stretch, more oxytocin.
Got it.
Oxytocin is therefore crucial for the powerful, expulsive contractions needed to deliver the baby in the second stage. And then in the third stage, after the baby is born, oxytocin plays a vital role in causing sustained uterine contraction to clamp down on blood vessels where the placenta was attached, preventing excessive postpartum hemorrhage.
So prostaglandins initiate and soften the cervix. Oxytocin sustains and strengthens contractions, especially for expulsion and after delivery, and it all feeds back on itself.
You've got it. It's a powerful self-amplifying cascade once it gets going.
Incredible. Okay, after delivery, the focus shifts. Another incredible physiological process begins, lactation. How does the breast actually develop to be ready for this, and what are the components of milk?
Right. Breast development starts at puberty, driven by estrogens and progesterone. But it's during pregnancy that the breasts undergo full development to become milk-producing organs. This is driven by the continued high levels of estrogens and progesterone, but also, crucially, by prolactin from the mother's pituitary and that human placental lactogen, HPL, we talked about.
What happens structurally?
The ductal system branches and grows, and the terminal ends develop into secretory units called alveoli. These are tiny sacs lined by milk-secreting epithelial cells and surrounded by a network of contractile cells called myoepithelial cells, plus adipose tissue. These alveoli are organized into lobules, which drain into ductules, then larger ducts, which widen into ampullae near the nipple, and finally open via the lactiferous ducts.
Like bunches of grapes, essentially.
That's a common analogy, yeah. Ready to produce and eject milk?
So what exactly is milk made of? And how is human milk uniquely suited for human babies compared to, say, cow's milk?
Milk is a complex and dynamic fluid. It's essentially an emulsion of fats suspended in an aqueous solution containing sugars, primarily lactose, proteins like lactobulin and casein, Various ions, sodium, potassium, chloride, calcium, vitamins, and immune factors.
And human milk differs from cow's milk.
Significantly. Cow's milk, for instance, has nearly 3 times more protein than human milk, mostly casein, and also much higher levels of electrolytes like calcium and phosphate. Human milk has more lactose and a different fat composition.
Why the difference?
It's tailored. A newborn human infant's gastrointestinal tract and kidneys are relatively immature and are specifically designed to handle the composition of human milk. The lower protein and solute load is easier on their kidneys, the specific types of fats are better absorbed, and human milk contains unique components like oligosaccharides that feed beneficial gut bacteria, plus vital immune cells and antibodies from the mother.
How do those alveolar cells actually secrete all these different components?
They use multiple routes, at least 5 major pathways operating simultaneously.
5 ways.
Yeah. 1. The main secretory pathway. Milk proteins like casein and lactalbumin are made in the endoplasmic reticulum, processed in the Golgi apparatus, where lactose is also synthesized, packaged into vesicles, and released by exocytosis into the alveolar lumen, the central space.
Okay, proteins and lactose out that way. What else?
2. Transcellular transport. Things from the mother's blood are taken up by the cell, transported across, and released into the milk. A key example is maternal immunoglobulins, especially IgA, which provides crucial protection for the infant's gut lining.
Passive immunity again.
Exactly. 3. The lipid pathway. Triglycerides are synthesized within the cell and coalesce into large lipid droplets. These droplets then move to the apical membrane, the side facing the lumen, and bud off, taking a piece of the cell membrane with them as they're secreted into the milk. That's how milk fat gets out.
Okay, proteins, antibodies, fats.
Yeah.
What about water and salts?
4. Transcellular salt and water transport. Electrolytes like sodium, potassium, and chloride are actively pumped across the cell membranes, and water follows osmotically, largely drawn by the high concentration of lactose synthesized within the Golgi vesicles.
Makes sense. One more?
5. The paracellular pathway. This is transport between the cells. The tight junctions connecting alveolar cells become a bit leakier during active lactation, allowing some water, salts, and even whole cells like maternal leukocytes, macrophages, neutrophils, lymphocytes to pass directly from the interstitial fluid into the milk.
Immune cells in the milk.
Yes, providing even more direct immune protection for the baby. It's an incredibly comprehensive nutritional and protective fluid.
Okay, so the breast is ready. The cells know how to make milk. What's the main hormone responsible for actually making the milk, driving that production?
That would be prolactin, PRL. It's a polypeptide hormone released from the anterior pituitary gland. Prolactin is essential for initiating and maintaining milk production.
Prolactin. And how is its release controlled?
Interestingly, prolactin release is normally inhibited by the brain, specifically by dopamine released from neurons in the hypothalamus. Dopamine acts on the pituitary lactotrophs to keep prolactin secretion low most of the time.
So to get milk production, you need to remove that inhibition.
Precisely. And the most powerful physiological stimulus for prolactin release is suckling by the infant.
How does suckling do that?
Nipple stimulation sends sensory nerve signals up the spinal cord to the hypothalamus. These signals inhibit those dopamine-releasing neurons. Less dopamine means less inhibition of the pituitary lactotrophs, so they release bursts of prolactin into the bloodstream.
So suckling directly triggers prolactin release.
Yes. And prolactin then acts on the mammary glands to promote mammary growth, mammogenic effect, initiate milk secretion after birth, lactogenic effect, and most importantly, maintain milk production once it's established, the lactopoietic effect. Keep stimulating, keep producing.
Is prolactin the only factor needed to start milk production after birth?
It's the key driver. But initiating milk production, lactogenesis, also requires the abrupt fall in estrogen and progesterone levels that happens immediately after the placenta is delivered. During pregnancy, high estrogen and progesterone actually block prolactin's full milk-producing effect on the breast tissue.
So the brakes come off after delivery.
Exactly. Prolactin levels are high during pregnancy, but the estrogen-progesterone brakes are on. Placenta out, breaks off, prolactin works, milk comes in.
Okay, so prolactin makes the milk, but how does it actually get out of the alveoli and ducts to the baby? Milk doesn't just flow freely, does it?
No, it needs an active ejection mechanism, and that's where oxytocin comes back into the picture.
Oxytocin again, busy hormone.
Very dizzy. Oxytocin enhances milk ejection. This is often called the milk letdown reflex. It does this by stimulating the contraction of those specialized myoepithelial cells that surround the alveoli and ducts in the breast.
So oxytocin causes those cells to squeeze.
Exactly. They contract, squeezing the milk that's been produced and stored in the alveoli down through the duct system towards the nipple. This is its galactokinetic effect.
And that's why nursing can sometimes cause uterine cramps, right? Oxytocin is contracting the uterus too?
Precisely. Oxytocin doesn't just target the breast, it also targets the uterus, helping it contract back down towards its pre-pregnancy size involution and reducing postpartum bleeding. So those after pains are actually a sign the system is working.
How does the letdown reflex work? How is oxytocin released?
Similar to prolactin, it's triggered by suckling. Suckling stimulates sensory nerves in the nipple, sending signals via the spinal cord to the hypothalamus. But this time, the signal goes to the neurons that produce oxytocin, causing rapid bursts of oxytocin release from the posterior pituitary into the bloodstream.
Okay.
When oxytocin reaches the myoepithelial cells in the breast via the blood, they contract, and milk is ejected. This usually happens about 30 to 60 seconds after suckling begins. The mother might feel a tingling sensation.
And I've heard that even just thinking about the baby or hearing a baby cry can trigger letdown. Is that true?
Absolutely. Unlike prolactin release, oxytocin release is highly influenced by higher brain centers. So yes, psychic stimuli, the sight, sound, or even smell of the infant can become conditioned triggers for oxytocin release and initiate the letdown reflex, sometimes even before the baby starts nursing. Milk might start leaking.
Wow.
However, the flip side is also true. Stress, fear, pain, or anxiety can inhibit oxytocin release from the brain, which can suppress the letdown reflex and make it difficult for milk to flow, even if the breasts are full.
So relaxation is key for breastfeeding.
Very important, yes.
Okay, one final piece. Does suckling and lactation impact a mother's fertility? Does it prevent ovulation?
Yes, generally it does, especially in the early months with frequent exclusive breastfeeding. Lactation tends to inhibit cyclic ovulatory function.
How?
The suckling stimulus, besides triggering prolactin and oxytocin, also appears to suppress the pulsatile release of gonadotropin-releasing hormone, GnRH, from the hypothalamus.
GnRH, that controls pituitary hormones.
Exactly. Less GnRH means reduced secretion of follicle-stimulating hormone, FSH, and luteinizing hormone, LH, from the anterior pituitary. And without adequate FSH and LH stimulation, the ovaries don't develop follicles properly, ovulation doesn't occur, and menstrual cycles are suppressed. It's nature's way of spacing pregnancies to some extent.
But it's not foolproof birth control.
Definitely not foolproof. Ovulation can eventually resume even while breastfeeding, often without a preceding menstrual period, so it's not reliable contraception long term. The timing varies greatly depending on suckling intensity, frequency, and individual factors.
Wow, what an incredible journey we've taken today. Seriously, from that microscopic dance of fertilization, the zygote's perilous journey, the intricate construction and function of the placenta,
The complete overhaul of the mother's physiology to support the pregnancy.
the powerful orchestration of labor, and then this whole other complex system of lactation kicking in. It's truly mind-boggling how all these systems work in such perfect concert, adapting, transforming, all to create and nurture new life.
It really highlights the immense physiological adaptations that occur. And understanding these complex mechanisms, it not only gives you, hopefully, a deeper appreciation for the human bowel, but it also provides absolutely essential context for diagnostics, pathology, and treatment in clinical settings. You can really see how the body prioritizes the continuation of life.
So what does this all mean for you listening? As a student of physiology, it means you've just gained, I hope, a profound understanding of one of life's most beautiful and complex chapters. It's a lot, I know, but don't feel overwhelmed by the details. Try to always connect them back to the big picture, the overall story, and the incredible processes they represent.
You are absolutely capable of mastering this material. We really hope this deep dive has given you some of those aha moments, clarified some tricky points, and boosted your confidence. Keep exploring, keep asking questions, and know that you are a vital part of the deep dive family.
Until next time, keep that curiosity alive.